Bismillahirrahmanirrahim. My name is Ammara Rauf, and today I am giving you a lecture on the digestion and absorption of the macromolecules, which is our first lecture. So we start. First of all, class we see the introduction of the digestive system. We also already have uh, seen that the digestive system is defined as the process by which the food is broken down into simple chemical substance. Like the food which is in the complex form is converted into a simple form, so under which circumstances it is converted into the simple chemical substances. The procedure which we involve here are basically the mechanical and chemical. We see on the complex carbohydrate, on the complex proteins, on the complex fats. During the process of digestion, different type of enzyme will act and they convert these substances into very simple molecules, so that these molecules can be absorbed to our blood. And when these molecules will absorb to our blood, uh, then through blood these substances will reach to the cell and tissues, where they provide energy. So digestion is defined as a process by which the food is broken down into the simple chemical substances that can be absorbed and used as the nutrients by the body. Then we see basically the function of the digestive system. The function of the digestive system include ingestion or consumption of food substances. What is basically the first function? If we see the digestive system, consumption of food substances means use of food substances. Then second function is breaking down into small particles. Then third function is transport of small particles to different areas of digestive tract, mean transport of the food particle to stomach, to small intestine, to the large intestine. And then there is the secretion of necessary enzyme and other substances for digestion, mean digestive system will release specific enzymes that will cause the breakdown of carbohydrate, proteins, fats, that are the macromolecule, and convert them into the micromolecules, so that these molecules will be absorbed to our body. Then digestion of food particle. Digestion is basically the conversion of macromolecule into the small particle. Then there is absorption of the digestive product is the main function of the digestive system, and then removal of the unwanted substances from the body. Remove कर देना unwanted substances को from our body is the all are basically the function of digestive system. Then we see the functional anatomy of the digestive system, which will include mouth, pharynx, esophagus, then there is stomach, then there is small intestine, and then large intestine, and then large intestine. After that there is rectum and anus. Then there is accessory digestive organs. These are basically the organs which assist in the digestion, but they directly not play a role, but they assist other organs. They assist the alimentary canal, you can say it as, like the teeth, tongue, the salivary gland which secrete saliva, the exocrine part of pancreas. The pancreas basically is a dual gland. It has endocrine properties and exocrine. So we see what is basically the endocrine and exocrine difference. Endocrine glands are basically those glands which secrete their secretion directly into the blood, while exocrine glands are those glands which secrete their secretion into the duct. So the which part of the pancreas is we will see in digestive system, we will see the exocrine part of the pancreas. The endocrine part of the pancreas we will discuss in the endocrinology. Then there is liver, and then there is gallbladder. Liver also secrete bile secretions. Gallbladder will store this bile and secrete when the intestine need it. That storage will be temporary. Gallbladder will store the bile which is coming from liver. Okay, so these are basically all the accessory organs: the tongue, salivary gland, exocrine part of the pancreas, liver, and then there is gallbladder. Then we will see the walls of GIT. The wall of GIT has 4 layers. GIT wall contains 4 layers. First is a mucus layer, second is a submucous layer, then there is muscular layer, and fourth is a serous or fibrous layer. Then these mucus layers, then submucous layer, muscular layer, serous and fibrous layer further divided into 3 and 2 layers. Like if we can say that the mucus layer is this, from this to this. It is contain epithelial lining, then there is, we see epithelial lining of the mucus layer. Then after that there is a submucous layer. This one is a submucous layer which I am scrolling, where arrow I am scrolling here. Then there is muscular layer. The muscular layer of the stomach and the small intestine has a small extent, there is a difference. The stomach has in the small intestine there is difference in the circular arrangement and the longitudinal arrangement, the inner circular and outer longitudinal muscle. Then there is the serous and the fibrous layer. Okay, then we can see the general organization of the GIT tract. All the layers which we discussed previously we can see here. As we say that the mucus, submucus, muscularis and the serous layer also further has a different type of smaller level layers. Like the mucus layer has a surface epithelium, basal lamina, lamina propria and muscularis mucosa. Then there is a submucous layer. Submucous layer के बाद we can see the muscularis externa, which is muscular layer. In the muscularis layer we can see there is inner circular layer and outer longitudinal layer, and then after the muscular layer there is serosa, which is serous layer, we already discussed in the previous here we can say that there are walls of the GIT has the mucus layer, submucous layer, muscularis layer, serous or the fibrous layer. And then we say that the mucus layer has further surface epithelium, basal lamina, lamina propria and muscularis mucosa. And then there is submucous layer, and then there is muscularis externa. Muscularis externa is basically the muscular layer of the wall of the GIT, which is the third layer, and third layer has further inner circular and outer longitudinal layers. And then there is serosa, then that is the serous layer, that was the fourth one. And in these, in between the muscularis and we can say there is the inner muscularis, there is a Meissner nerve plexus and Orbeck's mesenteric nerve plexus. Basically these plexes will involve in the contraction and the regulation of the blood flow in the digestive canal. Then there are two ways of digestion. We can say that there is mechanical digestion and chemical digestion. Mechanical digestion will involve the breakdown of food particle into small pieces without chemical change into them into different substances. Mean there will be in the mechanical no chemical digestion, no enzyme will involve. We just chew, tear, grind, mash and mix the food. While in the chemical digestion, we can see that change the chemical composition of food with the aid of digestive enzyme. The digestive enzyme will involve in this digestion. This, as we can say, the lipid is converted into the fatty acid, protein to individual amino acid and carbohydrate into the simple जो है वो sugar is converted in chemical digestion. So in this slide we see there are two ways of digestion. First is the mechanical and second is the chemical. In the mechanical digestion there is no chemical or no enzyme will involve in digestion of food. We simply chew, tear, grind, mash, mix the food. And in chemical digestion different type of enzyme, lipolytic enzyme will involve for conversion of जो है वो lipid into the fatty acid, as we can say the bile salts which are chemical and they will involve and conversion of the lipid into the fatty acid. Peptide is involved in the conversion of the protein into the amino acid. So in this slide we see mechanical and chemical digestion differences. Now mechanism of digestion at different in the previous slide as we see the mechanical and the chemical digestion. So mechanism of digestion at the different level from starting from mouth to the anus. The chemical and mechanical digestion will in both run, we can say it as run with each other. Like mechanical digestion include the chewing and swallowing at the level of mouth, and chemical digestion of carbohydrate and fats is start from here. Also the chemical digestion of carbohydrate and to some extent fat is start from mouth, because in the mouth different type of salivary gland that was submandibular, sublingual and parotid gland which gives saliva, and the saliva contain different type of enzyme which will cause the chemical digestion of carbohydrate and fats. Then we will see the mechanical digestion include peristaltic mixing and propulsion at the level of stomach. First we see at the level of mouth, now we see at the level of stomach. At the level of stomach there will be the mechanical digestion, there is chemical digestion. Chemical digestion of proteins and fats will take place at this level, and mechanical digestion with peristaltic mixing. Peristaltic mixing basically क्या होते हैं, that will be the wave like contraction and relaxation. That movement will move throughout the alimentary canal, we can say as. So at the level of stomach there is peristaltic movement, that will be in mechanical digestion. And if we say the chemical digestion of protein and fat will take place here, and absorption of the lipid soluble substances such as alcohol and aspirin. Then at the level of the small intestine, there is mechanical digestion, there is also chemical digestion, and chemical digestion of all the macromolecules including carbohydrate, fats and polypeptides, and the nucleic acid will take place in the small intestine, and also absorption of peptide, amino acid, glucose, fructose, fats, water, minerals and vitamins will also take place at the level of small intestine. So we see there is mechanical and chemical digestion. At the level of stomach there is the mechanical, chemical and absorption of some extent lipid, and at the level of small intestine there will be the mechanical digestion, there will be chemical digestion and also absorption of peptides. Then we see at the level of large intestine, mechanical digestion, and also to no chemical digestion will take place at the level of large intestine, and absorption of ions, minerals, vitamins, and organic substances will take place at this level. So we see the mechanism as in the previous. We see chemical and mechanical digestion, then at different levels, at the level of mouth, at the level of stomach, at the level of small intestine, at the level of large intestine, we see the mechanical and chemical digestion. So through this diagram we can also say that the digestion of carbohydrate will start from mouth, the digestion of proteins and fats will start from stomach, and lipid is the only one whose digestion will take place at the level of small intestine. Now we see what are the phases of digestive system. Ingestion occur when the material enter via the mouth, मतलब we take the food material. Then there is mechanical processing, crushing, shearing, tearing of a food. Then there is digestion which will chemical breakdown of organic substances. Secretions, different type of secretion from the GIT tract epithelium, and different type of gland will secrete their secretions. And then there is absorption, the movement of organic substances and electrolyte, vitamins, and digestive epithelium into the blood. There will be absorption will take place. And then there is excretion, removal of the waste product from the body fluid is excretion. So what are the phases? Ingestion, mechanical processing, digestion, secretion, absorption, and excretion is the phases of digestive system. Then mouth. Function of the mouth include ingestion of the food materials, chewing the food and mixing it with saliva, appreciation of taste of the food through tongue, transfer of food to the esophagus by swallowing, role in speech, social function such as smiling and other expression. And then tongue pushes the food to the back of the mouth, and what is the food material at the level of mouth is known as the bolus. And bolus, what is bolus? A soft mass of rounded ball of food that is being chewed, जो कि chewable है, soft mass में, soft mass form में, rounded ball of food है, which that is being chewed is known as the bolus. Okay, we see mechanism. Then we see the phases. We see them at the level of mouth, what is basically the function of the mouth. Then salivary secretion, we see here. Saliva, basically the mouth will also secrete with the saliva. The saliva is basically comes from the three type of glands: parotid gland, which is present at the front of ear; mandibular gland; submandibular gland; sublingual gland, which is below the tongue; and many small buccal glands in the mouth cavity. The pH of saliva is basically 6 to 7. During active secretion, it will be 8. Daily secretion of saliva normally ranges between 800 and 1500 millimeters. Means daily secretion of saliva is 800 to 1500 milliliters basically. Then we see the saliva in the saliva. The salivary gland contribution is how much? Like the contribution by the each major salivary gland: parotid gland is 25, submaxillary gland is 70, and sublingual gland is 5%. Basically that is not submaxillary, it is submandibular gland, not submaxillary. Okay, glands of salivation: submandibular gland, and then each type of gland has their specific type of secretion. We can say it as, like we can say the submandibular gland will secrete parotid gland will secrete entirely serous secretion. Serous secretion basically contain ptyalin enzyme. Ptyalin enzyme will cause the digestion of proteins. Then there is submandibular gland which secrete both serous and mucus type. Then there is sublingual gland. Sublingual gland also secrete both serous and mucus type. And then there is buccal gland which secrete only mucus secretion. Then there is primary secretion, ptyalin, mucus, and extracellular fluid. Submandibular gland. Submandibular gland has both serous and the mucus type of secretions. We can say there is acini. These cells has both mucus and the serous secretion. These secretion will come into the duct. So this is also the type of exocrine gland secretions. We can say the salivary glands are the exocrine glands. Then we see the composition of saliva. Saliva has the water 99.5%, then solid 0.5%. Then in the solid we can say there is organic substances and inorganic substances and gases, oxygen, carbon dioxide, nitrogen, inorganic sodium, calcium, potassium, bicarbonate. And then we can see also here the organic substances, which is amylase, maltase, lipase, liposomes, phosphatase, carbohydrates, and glycerine, and mucin, albumin, proline, rich proteins, lactoferrin, IgA, which is an antibodies. So blood group antigen, free amino acid, non-proteinous substances like urea, keratin, and xanthine and hypoxanthine, and different substances have the different role. Further we will in the next lecture, in the next slide, we will also discuss all these type of substances. Then function of saliva is the preparation of food for swallowing, appreciation of taste, digestive function, cleansing, and productive function, role in speech, excretory functions, meaning excretion of the different type of substances, and regulation of the body temperature and regulation of the water balance. We also know that the saliva also has a main role in the regulation of the water balance and the regulation of the body temperature. Swelling and deglutition, propelling of the food from mouth to stomach through pharynx and esophagus, and the swelling deglutination will include the buccal, voluntary, and then involuntary pharyngeal phase and the esophageal phase. Deglutination, if we see in the diagram, there is the pharynx, there is the closure of the closure of larynx, then the bolus will come again to the esophagus. Esophagus has the two esophageal sphincter, that is upper esophageal sphincter and the lower esophageal sphincter, and then the food will come into the esophagus, and through the esophagus, the lower esophagus sphincter, and the food will come into the stomach. Swelling and deglutination is the oral voluntary stage, and there is pharyngeal and the esophageal stage. Pharyngeal, that will be the involuntary, and esophageal stage, that will also have involuntary stages. We can say here the soft pharynx, nares, vocal cord, approximate epiglottis closes the larynx, and at this level the upper esophageal sphincter will relax and pharynx contract, peristaltic movement will start, and respiration is totally stopped. That will be the involuntary pharyngeal stage. And if we see in the oral voluntary stage, in the voluntary stage, food is rolled posteriorly by pressure of tongue upward. When we move the tongue upward, the food is forcefully by pressure will go into the posterior part of our oral cavity. Then esophageal stage, that is involuntary pharyngeal stage, and then esophageal stage. Esophageal stage में primary peristalsis. Primary peristalsis will cause the movement of food from pharynx to the stomach, and then secondary peristalsis. Secondary peristalsis from distension of the esophagus by food. Distension of esophagus by food, and then secondary peristaltic will cause the movement of food toward the stomach. We can see it as. Then stomach is divided anatomically into the fundus. We see here fundus, the body antrum, and then pyloric region. The main gastric gland of the stomach is present in this part, in the part of body of the stomach, and the main gastric gland will present at this part, that is body. Then we see the function of the stomach is the secretory function of the stomach and the motor function of the stomach. The secretory function of the stomach is basically two type of glands. We see in the body there are two type of glands which will involve in the digestion. That is oxyntic gland, and then there is pyloric gland. The oxyntic gland will secrete HCl, pepsinogen, intrinsic factor, and mucus, and pyloric gland will secrete mainly the mucus and gastrin. The mucus protect the wall of the stomach, and the gastrin stimulate the secretion of HCl from the oxyntic gland of the stomach. So stomach has two type of gland, that will be the oxyntic gland and pyloric gland. Pyloric gland will secrete mucus that will protect the wall of the stomach, and gastrin, and the gastrin will cause the stimulation of the HCl. And then there is oxyntic gland. Oxyntic gland has different type of secretions. We see in this secretion from the oxyntic gland. Oxyntic gland has further divided into the mucous neck cell. We see here the mucous neck cell, then the peptic or the chief cells, and then parietal cells. Obviously, if the gastric gland or oxyntic gland has different type of cell, then they also secrete different type of secretions. Like mucous neck cell will secrete mucus. The chief cells will secrete the pepsinogen, and parietal cell which will secrete the hydrochloric acid and intrinsic factors. So we see here the chief cell which secrete the pepsinogen, renin, lipase, gelatinase, urease, and parietal cell which will hydrochloric acid intrinsic factor of Kessler. Mucous cell will secrete the mucin, J cell will secrete the gastrin, serotonin, and there is enterochromaffin like cells which will secrete histamine. Okay. Chief cells, we see the oxyntic gland will secrete G cell, parietal cell, mucus cell, and pyloric gland will secrete G cells as pyloric gland, enterochromaffin cells, and enterochromaffin like cell. These are three cells which present in the pyloric gland, and the pyloric gland will secrete the gastrin, serotonin, and histamine. And these three type of glands, these three type of enzymes, we can say it as gastrin, serotonin, and histamine, will involve in the stimulation of the hydrochloric acid from the parietal cell. And then we see the chief cell will secrete the pepsinogen, lipase, gelatinase, urease, that will convert a different type of carbohydrate, act on different type of different type of macromolecules and convert into the, like lipase, it will cause the digestion of the to some extent lipid, and gelatinase and urease and pepsinogen which will act on the protein specifically. And pepsinogen is basically in active form. When the hydrochloric acid will come in contact with the pepsinogen, it will convert it into the pepsin, and then this pepsin will act on the proteins and perform different type of functions, different type of function of digestion, we can say it is. Now we will see in this diagram the acid, hydrochloric acid or the secretion. Basically we are now discussing stomach. In the stomach we say there is fundus, body, antrum and pylorus. Then we see the secretory function and the motor function of stomach. Then secretory function may we say two types of gland, that will be the oxyntic gland and pyloric gland. Oxyntic gland has further three types of cells: mucus cell, chief cells or oxyntic cells, which each type of cell has different type of secretions. That chief cell will secrete the pepsinogen, parietal cell will secrete the hydrochloric acid, mucus cell will secrete the mucin. Then there is also you have a pyloric G cell, enterochromaffin cell and enterochromaffin like cells, and also these also have a different type of secretion. After that we see the acid secretion in the parietal cell. In the parietal cell, this is basically the parietal cell of the oxyntic gland of the stomach. The parietal cell basically take the carbon dioxide from the extracellular fluid and water and form the H2CO3 carbonic acid. In the parietal cell there is enzyme which is carbonic anhydrase, which make the carbonic acid. This carbonic acid is further converted into the hydrogen ion and bicarbonate. This bicarbonate HCO3 negative will go into the extracellular fluid and chlorine will come from the sodium chloride. The sodium and bicarbonate form the sodium bicarbonate, HCO3 negative and sodium will combine with each other and form the sodium bicarbonate, and then chlorine which will come in the cell, and from the cell it is transferred into the canaliculus, where hydrogen which come from the acid and chlorine which come from the extracellular fluid to the parietal cell to go into the canaliculus. They combine with each other and form the hydrogen chloride acid. This acid make the environment of the stomach acidic. The basic mechanism of hydrochloric acid secretion, which I discussed in the previous, will also we can read it from here. Then secretion and activation of the pepsinogen. We also discussed that the pepsinogen is basically in activated form. It is converted into the activated form pepsin by hydrochloric acid. When the hydrochloric acid come in contact with the pepsinogen, it will be converted into the pepsin. And pepsin function as an active proteolytic enzyme in the highly acidic medium. Highly acidic medium में optimum pH level is 1.8 to 3.5. Then proteolytic enzyme means conversion of proteins into the amino acid. Then secretion of the intrinsic factor. We also see the parietal cell will also secrete intrinsic factors. Why this is more important? The intrinsic factors which are secreted by the parietal cell has a more important in the absorption of vitamin B12 from the GIT. The absorption of vitamin B12 from the GIT tract is basically taken by the secretion of intrinsic factor, and this if gastritis may lead to pernicious anemia due to deficiency of vitamin B12. If the absorption of vitamin B12 not take place, then there is pernicious anemia will occur. Okay, so in this slide we see the parietal cell which secrete the intrinsic factors. This intrinsic factor will involve in the absorption of vitamin B12, and the absorption of vitamin B12 is mostly involved in the formation of RBCs. If the absorption of vitamin B12 will not take place, then there will be pernicious anemia, which is the type of anemia. So if a disease or gastritis occurs, then it may lead to pernicious anemia due to deficiency of vitamin B12. Okay, now we see the gastric juice. Gastric juice, the full-on composition we can see here: water, which is 99.5%, and there is solid 0.5%, organic substances, pepsin, renin, gastrin, lipase, gelatinase, urease, and then other organic substances, mucus and intrinsic factors. And then inorganic substances, you can see here hydrochloric acid, which is the most important, then bicarbonate, potassium, chloride, phosphate, sulfate, calcium, sodium. These are also the inorganic substances in the gastric juice. Gastric juice is released from the stomach. Then the motor function of the stomach is the storage of food and mixing of the parietal digestion of food to form chyme. So the bolus, which come from the esophagus, is now become, after mixing, it becomes chyme. Slow inputting of the chyme into the duodenum will occur through the motor function of the stomach. After that, we see pancreas. Okay. But if we go behind, what type of discussion, what slides we discussed here? We see the function of digestive system: mouth, pharynx, esophagus, stomach, and then with that we also discuss the digestive organs, which is pancreas, liver, gallbladder. Now we will discuss this. So we see the walls of GIT, general organization of the GIT, mechanical and chemical digestion, mechanism of digestion, then phases of digestion, then mouth, function of mouth, salivary glands, saliva, then composition of saliva, then the function of saliva, swelling and degglutination, swelling, stomach, function of stomach, gastric secretions, oxyntic gland, pyloric gland, oxyntic gland, which type of secretion will secrete, then what type of oxyntic cells has cells and what type of secretions, then acid secretions, then secretion and activation of the pepsinogen, then secretion of the intrinsic factor, then the gastric juice composition, then the motor function of the stomach. Now we are discussing here the pancreas. Pancreas is basically a dual gland. I also already tell you that the pancreas, what are basically the dual gland? Dual gland means that it is exocrine and the endocrine secretion. Exocrine secretion will directly come into the duct, and endocrine level secretion will directly go into the blood, because these gland also has blood vessels which surround it, so which secrete the gland will secrete their secretion directly into the blood. Is endocrine function of the gland, and if this gland secrete their secretion away into the duct, this is the exocrine part of the pancreas. And here we will discuss into the exocrine part, and then this gland, this small duct will come into the main duct, and then this main duct will open away at the level of small intestine, and this small intestine level is duodenum. Okay, so which here pancreas are digestive gland that serve two functions: secrete hormone that regulate the blood glucose level, and secrete the pancreatic juice that neutralize the acid in the stomach. So pancreas is a dual organ having a two function. The endocrine function involves production of hormone like insulin, and the exocrine function that involves secretion of digestive fluid. We see here the pancreatic most important digestive juice because it contains enzyme for digestion of the most of the food stuff. The volume is 1500 ml per day, pH is 8 to 8.3, and pancreatic digestive enzyme is secreted through pancreas. We here see the pancreatic juice composition: water, solid, organic, inorganic substances. Then there is enzyme. The main enzyme is the trypsin, pancreatic lipase, and other organic substances, albumin and globulin, which are basically small proteins. Then there is inorganic substances which include sodium, potassium, calcium, magnesium, bicarbonate, chloride, phosphate and sulfate. These are all the composition of the pancreatic juice, and this pancreatic juice will come into the duodenum. In the duodenum there will be the first level, we can say, where the lipids are converted into the small fatty acid. So pancreatic lipase play a role in the digestion of lipid here. Also we can see here there are different type of enzymes which are secreted from the pancreatic juice. There are different type of activators also. There are different type of substrate and there are different type of end product. We can see here the trypsin, we see enzyme which is secreted in the from the trypsin. Okay, we are talking about trypsin. Trypsin is activated by the enterokinase trypsin, and it will act on the protein substrate, and the end product is proteases and polypeptides. Chymotrypsin is also activated by the trypsin, and it will be converted into the protein. Where will be the enterokinase trypsin? Enterokinase trypsin is basically also secreted away from the we can say the wall of the small intestine, and proteins is its substrate on which this chymotrypsin will act, and it convert the protein into the polypeptide. Then carboxypeptide is also activated by trypsin, nuclease, elastase, collagenase. These are all activated by trypsin, and they will act on the RNA elastic ligand, and they will form the end product in the form of amino acid, mononucleotide amino acid. Pancreatic lipase that will act on the triglyceride, and monoglyceride will produce. Cholesterol ester hydrolase. These is enzymes which are produced produced from the pancreas, and alkaline medium will activate it, and it will act on the cholesterol ester and convert into the cholesterol and fatty acid. Then we see the phospholipase A, phospholipase B, cholipase. These are also activated by trypsin, and then they act on different enzymes by different substrate like cholesterol ester, phospholipid, lysophospholipid facilitated action, and then the end product is phosphoryl choline and fatty acid. Pancreatic amylase also will act on the starch and form dextrine and malt.